Hi, I'm Nicole.
And I'm Anya, and we chose the PDGF signaling pathway. Our video number is 114.
We selected the PDGF signaling pathway. The PDGF signaling, aka platelet-derived growth factor pathways, play roles in many functions, some of which include embryonic development, cell proliferation, cell migration, angiogenesis, which is the formation of new blood vessels from pre-existing blood vessels, and is a necessary component of fibroblast cell division, which helps with clotting blood.
PDGF receptors, called PDGFR, are classified as being part of Class III receptor tyrosine kinases and can be found on the surface of cells. The signaling molecules themselves have both hydrophilic and hydrophobic portions, but are mainly classified as hydrophilic due to their protein structure, which contains many hydrophilic amino acids, and in turn make it soluble in water and unable to easily cross the cell membrane.
The PDGF receptors are primarily involved in cell growth, proliferation, and tissue repair. They play a large role in wound healing by stimulating fibroblast and smooth muscle cell division. The pathway or pathways they participate in are also essential for embryonic development and blood vessel formation. Dysregulation of PDGF signaling can contribute to diseases such as cancer and fibrosis.
PDGF receptor proteins work by acting as receptors on the surface of cells. When the signaling molecule, platelet-derived growth factor, binds to the PDGF receptor, PDGFR, it causes the receptor to undergo a change in shape, a process called dimerization. This brings two PDGFR molecules together, activating them, and allowing them to phosphorylate each other on specific tyrosine residues. These phosphorylated tyrosines serve as docking sites for various signaling molecules inside the cell. Once these proteins bind, they activate a series of intracellular signaling pathways, including the RAS-MAPK pathway, which controls cell division and growth, and the PI3K-AKT pathway, which promotes cell survival. The overall result is that the cell responds by proliferating, migrating, and differentiating critical actions for processes like wound healing and tissue repair.
PDGFR receptor proteins offer several advantages, including their crucial role in cell growth, tissue repair, and wound healing. They help regulate cell proliferation and differentiation, supporting normal development and regeneration. However, their activation must be tightly controlled. Excessive or uncontrolled PDGF signaling can lead to mishaps, such as abnormal cell proliferation, contributing to diseases. Additionally, mutations or overexpression of PDGF receptors can disrupt normal cellular functions, leading to pathological conditions.
The PDGF signaling pathway involves several key players. The specific signaling molecule is PDGF, which binds to its receptor PDGFR, a receptor tyrosine kinase located on the cell membrane. Once activated, the receptor undergoes dimerization and autophosphorylation, creating docking sites for intracellular signaling molecules. Chemomolecules involved in the intracellular response include phospholase C-gamma, phosphoinositide 3 kinase, and RAS-MAPK signaling components, which regulate various cellular responses.
The correct sequence of events in the PDGF signaling pathway is PDGF binds to the PDGFR on the cell surface. PDGFR undergoes dimerization and also phosphorylation. Adaptor proteins such as GRB2 and signaling molecules such as P13K bind to phosphorylated tyrosine residues. Activation of downstream pathways, including the REST MAPK pathway for cell proliferation and the P13K AKT pathway for survival and growth. Cellular responses such as proliferation, migration, and differentiation are triggered.
The PDGF signaling pathway is designed to promote cell growth, division, and movement. When PDGF binds to its receptor, the cell receives signals to proliferate, survive, and migrate key processes for wound healing, tissue repair, and development. The pathway also helps form new blood vessels and maintain normal cell functions. However, this response must be carefully regulated to prevent excessive growth, which could lead to diseases like cancer.
To prevent overactivation, the PDGF signal pathway is tightly controlled and eventually shut down. Here's how. Dephosphorylation. Enzymes called phosphorylases remove phosphate groups from PDGFR and other signaling proteins, stopping their activity. Receptor internalization. The PDGF receptor is pulled into the cell through endocytosis, reducing its presence on the cell surface. Receptor degradation. Inside the cell, the receptor is broken down in lysosomes, preventing further signaling. Inhibitory proteins, negative regulators, like sprouting proteins, block downstream signals to turn off their response. PDGF breakdown. The PDGF signal is degraded or removed, ensuring it no longer stimulates the receptor. These steps ensure that the cell only responds when needed and prevents uncontrolled growth that could lead to disease.
Imatinib, also known as Gleevec, is a well-known antagonist of the PDGF signaling pathway. It is an exogenous, meaning externally introduced, small molecule drug designed to inhibit PDGF receptor tyrosine kinase activity. Imatinib works by binding to the ATP binding site of PDGFR, preventing its activation and blocking downstream signaling pathways that promote cell proliferation. This makes it an effective treatment for diseases caused by excessive PDGF signaling, such as certain cancers, including chronic myeloid leukemia and gastrointestinal stromal tumors.
Imatinib is a competitive inhibitor that blocks ATP binding site of the PDGF receptor. By preventing ATP from binding, it stops the receptor from phosphorylating itself and activating downstream signaling molecules. This inhibits key pathways like the RAS, MAPK, and P13K AKT pathways, which are normally responsible for cell growth, survival, and proliferation.
Since imatinib prevents PDGF receptor activation, the cell no longer receives signals to grow and divide. This is especially important in conditions where PDGF signaling is overactive, such as in certain cancers. By blocking this pathway, imatinib slows or stops uncontrolled cell proliferation, leading to reduced tumor growth. This altered response helps treat diseases like chronic myeloid leukemia and gastrointestinal stromal tumors, where abnormal PDGF signaling contributes to cancer progression.
Thank you for listening.